Welcome everyone. While we wait that everybody gets in the room, we're going to play a short video about IUFRO.
Hello, my name is Todd Ramsfield and I'm the coordinator of IUFRO Division 7 Forest Health. It's my pleasure to welcome you to this webinar series. Before we begin the webinar, I'd like to take a few minutes to introduce IUFRO and to let you know how you can become involved. IUFRO is a global, non-profit, non-governmental, and non-discriminatory scientific organization that has been operating for over 125 years. There are approximately 600 member organizations spread across over 120 countries, and more than 15,000 forest scientists participate within IUFRO. IUFRO is open to all individuals and organizations that conduct research on forests or forest products. One of the primary activities of IUFRO is to promote networking and knowledge exchange in order to provide science-based solutions to global forest challenges. IUFRO is composed of 9 divisions that cover research on all aspects of forest science, including silviculture, physiology and genetics, forest operations, forest assessment, forest products, social aspects of forestry, forest health, forest environment, and forest policy and economics. Specific subject areas within divisions are captured within research groups. For example, within Division 7, we have 2 research groups, forest pathology and forest entomology. Research groups each contain working parties that are focused on more specific subject matter. Another major component of a IUFRO structure is task forces. Task forces are temporary and composed of interdisciplinary teams that are focused on specific emerging topics. This slide shows the composition of Division 7. As you can see, there are 2 research groups, forest pathology and forest entomology. Within these research groups, there are 11 and 14 working parties respectively. The working parties include research on all aspects of forest health. It's often said that working parties are the active heart of IUFRO, as these groups get together regularly for meetings focused on their areas of study, and you can see some photos at the bottom of the screen from pre-COVID working party meetings. Due to the global pandemic, we have had to postpone in-person meetings, but it is very important that networking and communications are maintained. Therefore, we have transitioned to online meetings and webinars. These meetings have lowered the barriers to participation in working party activities and will hopefully open the doors for more researchers. The current webinar series will take place over October, November, and December, with a webinar every other Wednesday beginning on October the 13th. As you can see, the webinars in this series are intended to be of broad interest to all researchers within Division 7. Note that the start time for each webinar will vary to accommodate the arrangers and speakers, but they will all be recorded and uploaded to YouTube for future reference. This is the 3rd series of webinars, and 2 other series have already taken place. The first occurred from September to November 2020, and the second from January to April 2021. The webinars have already been uploaded to YouTube, so you can go there to watch them if you are interested. We are hoping that a reduction in COVID will allow in-person meetings again in the near future, and 2 of the upcoming events that we want to bring to your attention are the All Division 7 meeting in Lisbon, Portugal in September 2022, as well as the Russian Regional Congress later in September of 2022. Further information for these conferences can be found on the IUFRO website or the websites of the meetings themselves. Finally, IUFRO is open to everyone involved in forest research. If your institution is a member, you are automatically an IUFRO member. For example, graduate students that are studying at a university that is affiliated with IUFRO are able to participate as full IUFRO members. In my experience, IUFRO is very welcoming, and hopefully you will be able to participate. Feel free to reach out to me. My email address is on this slide, or any member of the coordination team. We are easy to find on the IUFRO website as well, and happy to chat. Thanks, and enjoy the webinar.
Okay, well, I'm Caterina Villari with the University of Georgia, and I would like to welcome you all to this 3rd series of webinars organized by Division 7 and Forest Health within IUFRO. But before we go to talk about the specific session of today, I would like to introduce and thank our technical support for all this seminar series. So please meet Kyra, Lynn, and Felipe Balocchi. They're both PhD candidates within FABI in South Africa, and they're going to help us throughout all the session as technical support. So they will be in charge of meeting people, helping with questions, making sure every presenter is able to share the slide correctly, and so on. So please thank them, and I would like to again welcome them. Now, today's session. So this is the first of the webinar series, and the topic of today is tree resistant to diseases and pests, interaction with and facilitation by the microbiome. This specific session has been organized by myself together with Richard Sniezko, USDA Forest Service at the Caterina Genetic Resource Center, and Anna Nelson, USDA Forest Service Southern Research Station. And in particular, we represent the working party 70311, resistant to insect and pathogens, of which I'm the coordinator, and the working party 20215, breeding and genetic resources of the fine needle pines, of which Richard is the coordinator. And please know that this is this specific session is organized in together with the working party, with Division 7 and the Division 2, which is physiology and genetics. And now I would like to introduce you to the speakers. Dr. Kathrin Blumenstein with the University of Göttingen. She will talk about the Diplodia Tip Blight outbreak in Scott pine, influences of the mycobiome and abiotic factors. Dr. George Newcombe with the University of Idaho in the US, who will talk to us about pathogens in the trillion tree era. And Dr. Steve Wakelin with Scion in New Zealand, who will talk to us about Pinus radiata, a model tree microbiome system for a rapidly changing world. And I would like to thank them for connecting from different time all across the world. And the format of this webinar is that each speaker will have a presentation of 15-20 minutes, and then we'll have some time for questions specific to them. We will read the questions, so please write all the questions in the chat. If we're not going to have time to answer to all of them, we'll transcribe them and make sure that the speaker receives them via email, so that you will get your answer anyway. And then after the specific talks and the questions specific to the talk, we're going to have about 20 minutes of panel discussion moderated by Richard, in which we'll try to have a fruitful discussion about the topic of today. And with this, I would like to pass it directly to Dr. Blumenstein with her presentation.
Hello, everyone. And yeah, I'm sharing my screen. Yeah, hello everyone again. Thank you for joining today also from my side. And thank you, Caterina, for the kind introduction. My presentation is going to give you a short insight into the research of my group and collaborators. I assume Diplodia Tip Blight is a known disease for several of you. In Europe, this disease is increasing in the last decade, and therefore it's a highly relevant research topic here. The responsible pathogen has 3 equally used synonyms that are Thropsis sapinea, which is the synonym I'm going to use, and Diplodia sapinea or Diplodia pinea. And this fungus belongs to the Ascomycota, an anamorphic Botryospheriaceae. The specialty of this fungus is that it can perform a lifestyle switch. So it can occur as an endophyte, meaning it does not cause any symptoms to its host, and it can become a saprotroph or switch and become a wound and weakness parasite. The disease outbreaks happen and symptoms become visible when the trees are weakened by certain stress-causing factors, such as drought, especially. Generally, it prefers warm conditions and it's distributed via wind, water droplets, and insects. Here in the photos, you can see pycnidia on infected needles, and you see an opened pycnidium with a conidia, and here's fungal culture on malt yeast peptone agar. Thropsis sapinea infects preferably conifers and especially pine species. In Europe, the main host is Scots pine, which is one of the most economically important forestry tree species. But also Austrian pine, fir, spruce, larch, and Douglas fir are preferred. But lately the fungus has also been isolated from beech and black alder. The typical symptoms we can see are manifold. We can see shoot blight, shoot deformation, browning of the needles, light grounds due to reduction. In the inner wood, we can see blue staining, bark lesions or cankers, and on the entire tree, crown wilting or dying of entire trees. And now we come to the study that I like to introduce you to. So it is a collaboration between my group at the University Göttingen and the Federal Research Institute, also located in Göttingen. This study is published in Frontiers. Here's the title. And we were interested in investigating the pathosystem of Thropsis sapinea and Pinus sylvestris. So first we wanted to identify fungal Scots pine endophytes, which may have the ability to antagonize the pathogen in vitro through a dual culture screening. Second, to test how the strength of drought-induced stress due to different levels of water availability to the host affects potential disease symptoms. So measured by necrosis lengths, in inoculation tests performed with Thropsis and selected Scots pine endophytes in a greenhouse experiment, so in the living plant. And third, to test for potential remote synergistic or antagonistic influences between the Scots pine endophytes and the pathogen, also in a greenhouse study. In order to answer our first task, to find endophytes that may antagonize the pathogen, we performed antagonism assays. It's also called dual culture or paired growth assays. The fungi that we used were a set of 27 endophytes that we regularly isolate from pines in Germany. Also, in previous studies, we find those fungi more or less every time as the most abundant ones. And we used those 27 against 2 Thropsis isolates. So here the 2 fungi are placed on opposite directions with a defined distance, as you can see on this figure. And we measured along the alpha and the beta axis after 3, 7, and 10 days. And every combination was prepared with 3 technical replicates, and the fungi grew at room temperature, which is about 20 degrees frost, and no special light requirements. The ability to antagonize the pathogen was based on inhibition level. So for this, we grouped the results in 4 categories. Here I have some examples for you. So the pathogen is always on the right side and the endophytes on the left. The first category is the inhibition of Thropsis growth. This may happen due to chemical antagonism when the fungus produces a secondary metabolite that it releases in the agar around its colony. Second, when the endophyte is superior in the growth and when it has overgrown the mycelium of Thropsis, which may imply mycoparasitism. The fungus has either a stronger capacity to metabolize the nutrients or faster, which is an advantage during competition. We defined the third category as equal growth capability when the tested endophyte and Thropsis did not overgrow or did not obviously inhibit each other, so meaning neutral or no interaction. And here in the fourth category, Thropsis is superior, so it inhibits and may also have overgrown the tested endophyte strain. So this is the loser endophyte group. We evaluated the results of these antagonism assays in 2 ways. So the visual observation that I just showed you and statistically based on the measurements. And we found that 50% of the tested strains inhibited or were superior in their growth towards Thropsis, which means 13 potential antagonists. So here, for example, Pseudoia polysporum and the Aporta species, both with an inhibition of the pathogen growth, and Xularia polymorpha showing a superior growth towards the pathogen. And all 3 are very abundant endophytes in Scots pines. So meaning, in theory, a strong competition against the pathogen is possible. So I'm going to come back to this table in a bit. Now to the part of the study that we performed in a greenhouse. So as we wanted to test how the drought-induced stress affects potential disease symptoms, we arranged 2-year-old Scots pines in our greenhouse, 1008 little trees grouped in 4 watering treatments. So 100% being considered as the optimal amount, when 25% of that amount keeping the trees just alive. But let me tell you, they were really super drought stressed. And with 50% and 75%, we had 2 intermediate levels. Additionally, to just the infections with the pathogen, we wanted to see if there are potential remote influences, such as synergistic or antagonistic effects between the pine endophytes and other pine pathogens and Thropsis. So we compared the infection capacity of these fungi both alone and in combination infections. For this, the tip of a side shoot was cut off, like 1 centimeter, and replaced by an agar plug that was inoculated with the respective fungal mycelium and wrapped with parafilm to prevent from drying out. So which fungi did we choose? So here we are back now at this table. First, we decided to include a fungus with an unspecified status to serve as a neutral control, so to say. That was the Dimelaceae species. Second, we chose for Pseudoia polyspora, that is an abundant pine endophyte that can also become a pathogen, and here to test for remote antagonistic effects. Third, Microthropsis olivacea. It's a plant pathogen and quite a common fungus in conifer species. So here we were curious for a synergistic effect on the host. As the fourth fungus, we included Truncatella conorum piquea, a pine endophyte, and secondary pathogen, also here to test for remote synergistic effects. And the last one we included was Desmasirella acicola, an abundant pine endophyte, to test for antagonistic effects. So here you see again the overview. So we infected a set of trees from each water treatment group with each of these fungi in single infections, so only 1 tree infected with 1 of these fungi, and in combinations of each time 4 of those. And of course, we also included a control plug with only agar. Yeah, the results. So after 66 days, we ended the experiment and the trees looked like this here. So one side shoot infected with Thropsis. So they were mostly dead, as you can see on this one here with the red tag. And this graph shows the necrosis length per shoot for those trees where only 1 shoot got infected. All untreated control plants stayed healthy during the experiment. And in general, the twig mortality was highest in all the 25% water group compared to the 100% water group. So the fact that several endophytes or other pine pathogens did not cause higher necrosis rates may be due to the fact that those fungi perhaps were already present in the host by being typical twig endophytes, and Pseudoia polyspora and Truncatella conorum piquea, they did not cause strong necrosis on the twigs, probably because they are specialized true endophytes of Scots pine with a balanced host fungal continuum. So in this study, we did not aim at directly inhibiting Thropsis growth, but testing for an influence, causing like low necrosis, for example, as a consequence of these combined infections, like, for instance, an induced systemic resistance due to the presence of an endophyte. And the co-infections of the studied Scots pine with different endophytes and Thropsis on the different twigs did not cause increase in the plant's immune system, because we found the same necrosis development from the single infected to the combination infected trees. And the other fungi with the potential to become pathogens, they did not benefit from the host stress in this setting. Regarding the methodology we used and what can be applied in future studies is, for example, changing the inoculation method. So the inoculation with mycelium plugs works really well it's very reliable and effective, but it is also a quite aggressive method. So more natural is than using Conidia spores for the infection. We've also done experiments with the spray infections, and it works also really well. But the preparation of the spore solution is really time consuming and requires a lot of work. But it is, of course, more natural. And to my mind, one needs to decide where the priorities in the experimental setup are. Something else that can be changed in future studies is perhaps to first inoculate the trees with a respective endophyte, let them get established in the host tissues, and then followed by infection with the pathogen. And third, for this study, it was totally irrelevant to have 50% and 75% water group. And now in our latest greenhouse infection studies, we have only applied 60% water group. That seems to be efficient. So in conclusion, Thropsis has a high growth rate, indicating it has a fast metabolism to use available nutrients. The necrosis caused by Thropsis was statistically higher in all the water treatments compared to the other fungi, indicating that the host trees can particularly benefit this opportunistic pathogen. The different modes of the competition between the endophyte and Thropsis in the dual culture tests, they may indicate that other endophytes in the pine tissues influence the disease development of Diplodia Tip Blight. But when the pines are weakened due to drought stress, the common endophytes could be disadvantaged, while Thropsis can become more established and occupy more tissues faster, outcompeting inferior endophytes. So finally, I like to thank you for your interest, and I like to thank my team, which you can see here. Each of them has contributed in one way or the other, and special thanks to Dr. Eva Terhunen, who has been head of the group until half a year ago, and also to our collaborators from the Federal Research Institute, Dr. Lange and Dr. Buskamp. And here are two publications from my group and colleagues from this year, especially related to this topic. So this study where we investigated pine mycobiomes in regard to natural infections with Thropsis with different health status, and another study on the virulence of the strains from various hosts of this pathogen. And yeah, here's my email address in case you like to send further questions. Thank you very much.
Thank you very much, Kathrin, for your presentation. Do we have any questions? Please feel free to write them in the chat, and we will read them aloud and have Kathrin answering. Yes, we have full 5 minutes still, so that's good. Anyone wants to go with a question? Well, I have one.
Go ahead, Kathrin.
Oh, sorry. Okay.
It's Todd.
Oh, hi, Todd. Yeah, go ahead. You can go ahead.
Kathrin, that was a very interesting talk. Thank you very much. I find it really neat. You've got one endophyte / pathogen and then a whole bunch of other things within the tree. And you may have answered this a little bit in your table, but in your fungi that were superior endophytes, some of those could also be endophytes and switch to a pathogen lifestyle as well. So do you worry that your potential control for Thropsis would actually result in some damage to the host as well?
Yeah, that was one of the questions we were really interested in this study. So we were not only aiming at the antagonistic effect, as I mentioned, also if there are already testing for synergistic, as now with regard to climate change, we can see that several fungi have the potential to switch, becoming pathogens. So not only Thropsis subtinea, and this is something we really need to be aware of, and like know your enemies. We need to know which others can have this capacity and have them better under observation, like the Sedoia polyspora. That is one that is really abundant in the pines, like the second abundant one, and that may become also pathogenic. So I think further tests like this can be really useful.
Yeah, well, yeah, I really like the system. Thank you for sharing. So.
Looks like we have a question from Brenda or Mike.
Yeah, you see, I'm trying to fool you. You don't know who's going to be speaking.
Yes.
Two people in one house in 2 different rooms. Nice to see you all. And Kathrin, thank you so much for a really interesting talk. And I hope you don't mind me, Caterina, asking a question verbally. I hate typing in the chat box, and I type very slowly. So the talk will be over by the time I type. So you raised the issue of wound inoculation versus inoculation with spores. And I mean, it's just a fascinating part of the whole biology of this pathogen. I call it Diplodia sapinea. I'm going to ask you that question later, maybe on an email, why you chose that name. But yeah, you know, we know that the pathogen exists in the trees and expresses itself on trees under stress without wounds. In fact, I don't think it really is in nature mostly a wound infecting pathogen. The question, and you raised that, is how does one bring that biology into what you're doing? If you spray spores onto trees, you're going to get symptoms. But what, you know, is there a way that one could pre-inoculate trees so that the fungus is living within the trees and then start to understand what these other organisms are doing? I don't know how to do it, but it's something we need to think about. It's incredibly complex. And I don't know what the answer is, but you know, given that that's the way that Sphaeropsis is existing in nature, in some way we've got to come up with a system of pre-inoculating plants and then testing the effect of these other organisms.
Yeah, I remember that you've had this discussion before after the conference in Florence. We discussed about exactly that.
I'm sorry to raise it again. I guess
no, it's still relevant, and we've actually done some studies that is like 2.5 years ago. So we've done lots of studies with these spray infections because we really want to have a more natural infection method in order to be able to answer exactly that question. And so far, we found out that when we just spray these conidia on the trees and do not cause any injuries or cuttings, nothing happens. So the fungus does not seem to be able to enter the tree. We tried different methods, and but when we injure the trees to mimic like insect bites or hail or these kind of disturbances, then we do get pretty good symptoms. Yeah, so we are still on it because we want to mimic more natural infection ways to study this system. But as I said, yeah, with the mycelium, it's so much faster and easier. So it's a trade-off. Yeah.
Yeah, it's tricky. I think some of the molecular genetic tools maybe will enable us to understand better. We need to think some. But thanks. That's great.
All right. Well, thank you. We have 1 minute left for one other question, and then the other one we can read them through the panel discussion. So I have a question from Jeff Hamilton. He says, you mentioned beech trees as being affected. Are there any other hardwoods that may be affected?
Yeah, so far we also found it on black alder. Our colleagues from Research Institute actually isolated that. Yeah, so far that's what has been found. But I think if these two species are now also possible hosts, then it's just a question of time, I guess, until we find others as well.
Perfect. Thank you very much. Well, we'll read the other question during the panel discussion. There's some very interesting. I already see them, but we'll leave space to the next speaker, Dr. George Newcombe. Can you please share your screen and start your presentation? And please remember to unmute yourself.
And share my screen. Is that good?
Perfect. Yes, perfect.
Okay. So I chose this title because I've been thinking about the trillion tree era and thinking that if we're to add 1 trillion trees to the world's existing 3 trillion, and those are the estimates of Crowther et al. From 2015, that there's bound to be a call in the future, the near future, hopefully, for forest pathologists to help with some of the problems that will be augmented by all the tree planting. And let me see if I can advance my slide here. And of course, planting 1 trillion trees is not enough, or at least most people think that it's not enough to really stop climate change, but planting 1 trillion trees could be part of the solution. And of course, tree planting is something that peoples all over the world are engaging in because there are benefits in terms of water and shade, surface temperatures, and diversity, of course, not to mention food. And so I'm taking seriously the idea that we are going to plant 1 trillion trees. I'm not sure if it's at the rate of 100 billion per year over the next 12 years, accounting for a loss of 15 billion each year. But I'm assuming that there's going to be a lot of tree planting. And I've looked at my 30-year career as a plant pathologist, mostly as a forest pathologist, and tried to pick out highlights that might be applicable to 1 trillion tree scenario where a solution is needed that's fairly practical, that may be in a tree species that's never been grown before in a plantation. But I'll get to that in a few slides. I'm going to start with some of the papers that I published during the 1990s when I was working in hybrid poplar plantation forestry. Hybrid poplar in the Pacific Northwest of the US is based on interspecific hybridization with Populus trichocarpa. And when I got involved in this in the early 1990s, there were diagnostic issues to deal with. There were introductions of exotic Melamsera species that in one case, one exotic species hybridized with the native poplar rust, Melamsera occidentalis, and that released all sorts of new pathogenic variation. And the approach through all of this was to try to breed for resistance or select for resistance clones that might be able to grow through a full rotation without disease. And so there were a variety of problems that came up. We had geneticists involved, Reini Stetler at the University of Washington in particular, but there was also a commercial breeder, Brian Stanton, who was involved. And I worked on these problems in that setting. And towards the end of the 1990s, I realized that there was a pattern in the genetics of resistance to diseases of populace, that the exotic poplars were bringing in resistance that would show up in the hybrids. And unfortunately, the implication of that is that the native tree was bringing in susceptibility, but the breeders did move to totally exotic poplars, which are now really quite resistant to all the diseases of Populus trichocarpa. And that involvement in plantation forestry and genetics of resistance led me also to thinking about endophytes. Towards the end of the 1990s, there was variation in disease severity that wasn't explained by specific major genes for resistance. There were clearly lots of endophytes in populace, and so we started working on endophytes, and in particular on what we called the defense hierarchy in poplar. And this was work with Posey Busby, who's now an assistant professor at Oregon State. And we were curious not just about genes for resistance and endophyte-mediated resistance, but also about competition. In this case, you can see that the areophyid mite, and I'm not sure if you see the pointer moving, hopefully you do. This bronzed area is caused by an areophyid mite, and this area doesn't get much rust, versus areas that aren't bronzed that do get quite a bit of rust. And we call this pathogen competition. And here's the areophyid mite emerging from the stomate. It's quite an interesting creature. It was a new genus and species at the time, and it's endemic to the west side of the Cascade Mountains, or the maritime Pacific Northwest. And this is the hierarchy that we came up with genetic resistance at the top, in the top left, where if there's a major gene for resistance, you get necrotic flecking, but you don't get full development of uridynia. In the absence of the gene, the genetically susceptible state looks like this. This resistance is at the top in the sense that if there is a gene for resistance, then you're not going to be able to see pathogen competition, which is this middle photo. And you're also not going to be able to see endophyte-mediated resistance. You can only really see endophyte-mediated resistance, or we only saw it in our field experiments when we had genetically susceptible poplars and no pathogen competition. And then we could see significant endophyte-mediated resistance. And it's a little off here with what I'm choosing to represent endophyte-mediated resistance, because that's actually Eudarluca phylum, which is a mycoparasite and not really a endophyte. But anyway, that's what's standing in for endophyte in that photo. So we have this defense hierarchy. That's the first point I wanted to make. The second point that I think might be useful in the trillion tree era has to do with which endophytes to select. Assuming you're in a situation where there are no genes for resistance, you're working with some tree that no one's ever worked on before, you don't really have time to develop genetics, so you consider endophytes. Which ones do you look at? There are foliar endophytes, root endophytes. In this case, we're looking at seed endophytes, and we were doing a study of Centaurea stobi, the herbaceous invasive plant in the Pacific Northwest that we happened to sample to a great extent, both in its native range in Europe and Asia, as well as in North America. And we saw a really interesting pattern from thousands of seeds and thousands of isolates that either there was no culturable endophyte in a seed, or there was one, but there was almost never more than one. And we wondered about why that was. And so that led us to do experiments where we deliberately tried to infect seed of Centaurea with more than one endophyte. So we'd put 2 or 3 endophytes into flowers and then try to re-isolate the inoculants from the matured seed from those flowers. And what we found was that we could not do it. In fact, all we ever got was 1 endophyte per seed, and it was identity specific. If we co-inoculated endophyte A and endophyte B in a flower, A would always beat B, and A would be the only one we got out of that matured seed. Whereas if we did B and C, B would always be the one that we retrieved, or A and C, maybe C would be the one that we would always get. So these were completely exclusionary, therefore very strongly antagonistic interactions. And we had never really seen anything like this with foliar endophytes. So we thought we were onto something in terms of strong endophytes. And we even a few years ago hypothesized about a bottleneck in the plant microbiome in the seed, and the possibility that the primary symbiont, the one that was in that seed and was in such strong presence that it was the one that we cultured, it would have very strong effects on what happened to that seedling. And this has always struck me as important in thinking about trillion tree scenarios, because seedlings and saplings are the vulnerable stage in a tree's life. And so it made sense to me to think about these seed endophytes that had very strong effects. And we have documented that those effects are really quite strong in a number of scenarios. This was a case where I collaborated with Ray Calloway, who's an ecologist at the University of Montana, and we put in primary symbionts or seed endophytes into Centaurea, and he forced it to compete with North American grass species. And it was clear that the seed endophytes, primary symbionts, really contributed to plant-plant competition. But we've also done other things with primary symbionts, where in one case, Mary Ridout, working on her PhD at the time, showed that you could get recruitment effects from the primary symbiont. So she worked with one in wheat where the symbiont, the primary one, recruited dark septate endophytes that are useful in winter wheat over the winter. It recruited it to a much greater extent than other symbionts or nothing. And this is a very brief narrative, so I'm nearing the end here. Thinking again about the trillion tree scenario, and I've supposed throughout this that local groups trying to improve their environmental situation are likely to turn to local trees and to trees that really have no research basis or have not been researched to any great extent. And I thought I should investigate that a little bit just to prove to myself that there are trees, there are 60,000 tree species in the world. And I was assuming that many of those were not in the poplar, pine, eucalypt group, but instead in a group of trees that are largely unresearched. And so here's Cordia lutea, which is unresearched apart from one host pathogen record in the USDA BPI database. Or here's Sarcalina multiflora with no records at all of any fungal pathogens. And here's Hopia chinensis with some research. There's a chloroplast genome sequence for it, but no records in terms of pathogens. So it does seem plausible to me that in my hypothetical trillion tree scenario that people will want something that they could use like an endophyte, but they're going to want to know which endophyte. And they're going to look for something that will get their seedling through the vulnerable stage and get it established. And I know this is all very hypothetical. I'm assuming that there'll be lots of questions for this. I have already acknowledged many many people who have collaborated with me over the years in the references, but there are some others here. I wanted to mention in particular Abby Ferson. Her name is here. Abby is a current PhD student who's reached out to Richard to talk about experiments that we could do in white pine and porterfruit cedar and Acacia koa in Hawaii as well, to work on these primary symbionts that are seed endophytes, and in particular seed bacteria. Posey and I have a paper coming out soon, which is on the fact that you can isolate bacillus species that are really quite strongly antagonistic from seeds. The seeds are quite a good source. And I think I'll stop there. So I should stop share.
Yes, thank you very much. Very, very interesting talk and lots of idea behind it. So let's see. I already read a couple of questions. I think, well, the first one is more of a comment. In vegetable seed production, seed can be coated with biocontrol agents. This is from Ki Zhang. Is there?
Should I address that now?
Yes, please.
Yeah. Yeah. The thing that appeals to us about the fact that we're finding bacillus species in seeds is that bacillus species are already the basis for biopesticides. So it's not something that we feel we have to prove all that much, but we have been doing some antagonism assays and showing that these seed bacilli are really quite antagonistic. So we're expecting that they could be the basis of seed coatings in the future that people could use, given that there are already bacillus that are the basis for seed coatings.
Thanks. All right, we have a question from Mike. It's phrasing you that what you described is very fascinating. He says, in my experience with various endophytes on, say, pine, are not that vertically transmitted, but clearly horizontally. Certainly, there is true for, say, Diplodia sapinea. I like the idea of trying to determine what endophyte, and one that is associated with vertical transmission via seed makes a lot of sense.
I could just say that what we tend to see is, or what we've seen so far, is that seed endophytes tend to be not very common in foliage. And the endophytes that are dominant in foliage tend to be totally absent from seed. There's really not much overlap.
Can I butt in?
Sure.
If you have a few seconds, George, I mean, I'm so pleased you're looking at the endophyte situation from this perspective. You know, a lot of our literature is to do with grasses, which are very different. You know, you know, that question, for instance, from Kezang about trichoderma, for instance, where we've inoculated many times trichoderma to seeds. I mean, it has an effect on growth and what's in the root system, but one doesn't re-isolate it in the plant itself. So it seems, again, not to be vertically transmitted. Whereas, I mean, you mentioned some of these bacteria that clearly must be, and that makes them really interesting. And there's also the question, what are all the other fungi doing that are there? But that's another complicated issue.
Yeah. And I think, you know, it may be that primary symbionts don't directly affect pathogens later on in the life of a sapling. But they've had an effect in terms of recruitment, because the microbiome changes as a plant ages. Anyway, it's definitely a complex problem.
Well, we have 1 minute left. Let's read another question, and then we can keep discussing this during the panel discussion. So we have a question from Mohammed Suweri. Says, Dr. George, thank you for your clear presentation. Could you explain, handle, or control about incidents of root rot on eucalyptus forest plantation? In Indonesia, I have experienced finding some clone have damage by root rots like Ganoderma or Felinus.
That was Indonesia.
Yes.
Yeah, but that's a little bit beyond my experience, but and I'm assuming this is older trees.
I don't know, Mohammed, perhaps you can unmute yourself and explain a little bit better, and then we'll wrap it up and leave it to the last speaker. Well, we can leave this perhaps later on for the panel discussion.
Okay.
And in case Mohammed probably is not able to read or able to unmute himself. Okay, well, thank you very much, Dr. Newcombe, and
Yeah.
we'll call our last speaker, Steve Wakelin. We have to particularly thank because it's 3 a.m. where he is connecting right now, so this is really dedication to IUFRO meetings. Can you please share your screen and unmute yourself?
Always with the unmute.
Yes.
Every time. Have I shared the right screen, or is the screen got
Yes.
the notes on it?
No, this is the perfect one. Thank you.
Right, so good afternoon, everybody, and thank you very much to the organisers and conveners for inviting me along to talk this afternoon today, about some exciting new work that we have got currently underway here in New Zealand. It's in the area of the Pinus radiata, a model tree microbiome system that we hope is really going to help trees and forests into what is really a rapidly changing world. So I think we should start by admitting that forests are in quite a bad place globally, a very bad place. Climate and ecological breakdown are happening at terrifying speed, and certainly at a pace that far outraces nature's natural ability for forests to adapt and respond. And it's more than just the forests, of course, that are at stake. It's as much about the trees, the forests are about the water, the soil, communities, businesses, local and regional economies. And protecting our forests really means we might just have that chance to keep within safe planetary boundaries, you know, keeping safe ecosystems and ecosystem functions for a future for all of us and for our children and their children. So diseases are devastating forest ecosystems globally. It's not just illegal logging and land use change or deforestation that is impacting forests. It is pest diseases and other things such as those. And I suppose towards the top of our attention are pests such as the pine bark beetle. And this rather innocuous looking invertebrate is impacting large tracts of forests in the US and elsewhere. The impacts of this are particularly bad when both the beetle damage and drought are occurring simultaneously. And when these do occur together, entire stands of forest can be just outright killed. Other areas are not off the hook. Rather more striking emerald ash borer is a good example. This is a devastating pest of ash trees and is slowly marching west across Europe. And a particular concern will be when the range of this pest inevitably overlaps with that of other diseases such as ash dieback or cholera. So ash dieback alone has a tree mortality of something like 70 to 85%. And the combination of this disease and the borer pest may possibly spell the end of ash in many parts of Europe. In New Zealand, where I'm at the moment, the situation is no different. And although we are a geographically isolated country down the end of the world, we still somehow remain under siege from a constant barrage or an invasion of new pests and pathogens. It's likely that some of these new diseases are emerging as existing or sleeping pathogens that are created via increased mobilization of pathogenicity elements within microbial populations, or perhaps shifts in environmental conditions that release an organism into a pathogenic state, or maybe make the trees or forests more susceptible to those. Recent examples that we've had of new incursions have been myrtle rust, seen here on the left, and Kauri dieback disease, which is in the right panel there. And the Kauri dieback disease is particularly concerning in New Zealand. It's caused by a soil inhabiting root infecting Phytophthora oomycete, and it is literally killing off our native kauri forests. And these ecosystems can be ancient. You know, they're just getting started at 300 years, and trees that are thousands of years old are not uncommon. So these ecosystems are ancient. They hold immense cultural value and are home to significant native and endemic biodiversity in New Zealand. So these trees, which, you know, like I was saying, can be thousands of years old, can be devastated within decades of Kauri dieback. And as commentators in Nature have put it, this disease may very well be calling time on New Zealand's oldest tree species. And it's going to be a real shame if that keeps playing out the way it is. And of course, the other major threat is climate change itself. While many plants have been around for millions upon millions of years and have coexisted, moved, adapted, evolved and survived through glacial and interglacial periods, these events have largely occurred over very, very long time frames. And in this figure, we have a plot of global temperature with degrees Celsius on the y-axis. And I suppose on the x-axis, we've got time. But what's important to note there is that that's not linear. On the left-hand side of the panel, the time steps there are tens of millions of years, moving to what hundreds and tens of thousands of years in the middle panel. Yet it's in the next decade to hundreds of years that it's increasingly likely, given lack of urgency and action, that we will see more climate change than the planet has seen perhaps for the last millions of years. And for slow growing and sessile species such as trees, this isn't just climate change. It represents rapid alteration and often devastation of the biophysical conditions that these ecosystems can exist within and function within. So the key is that the rate of change that is occurring in the world and that the impacts are occurring simultaneously across biotic invasions, pests and diseases, and abiotic threats such as climate change, it's the rate and combination of these that is really devastating forests globally. Furthermore, it's unlikely that we can use tools such as conventional breeding approaches to escape the mess that we're in. We lack the time, we lack the tools, and we lack the knowledge, or all of the above, I suppose, to breed our way out of a lot of these problems. There's simply too many species involved, and we know too little about them, let alone their propagation and ways to manipulate them genetically. So essentially, the rules of the game have changed, and therefore we must find entirely new solutions to these issues. And what's really paradoxical is that at the same time that forests are under increasing threat, a growing population and transition to the biocircular economy means that the demand for wood and forest products is increasing all the time, expected to double by 2050. So not only must we find solutions for our forests, we must also find solutions for the products and things that we need from our forests. So potential opportunity to protect our forests from abiotic and biotic threats, altering their phenotype and fitness, is by harnessing their second genome, the microbiome. So human models have shown just how profoundly microorganisms that are present in and on our tissues influence nearly all aspects of our health and well-being. The microbiome in a human is malleable. It offers new opportunities not only to understand the pathophysiology of diseases from Parkinson's to autism and chronic fatigue, but to offer new solutions towards their treatment. And clearly, and the talks today have shown that plants also have their own microbiomes, both on their existing surfaces and also within their internal tissues. And as for humans, these plant microbiomes are intricately and intimately involved in plant health. They serve as reservoirs of entirely new genes that the plants may access and call upon to improve fitness, survival, and therein ecosystem stability and function. What we urgently need are a range of model systems for tree microbiome research where we can rapidly, robustly, and reproducibly assess the potential for management of the microbiome to protect trees and forest ecosystems. And it is as much as that we just can't rely on one single animal model for all of our needs. We haven't got just Drosophila or Xenopus, we've got a range of animal models. We also need the same for the tree microbiome systems. So here in Scion and in New Zealand, we are currently developing a tree microbiome model system based on the conifer Pinus radiata. Radiata pine has a number of wonderful attributes which leads itself really well to microbiome research. It has quite a fast growing cycle compared to many trees. We already hold really deep fundamental understanding of its physiology, ecology, and genetics. We have available a really rich infrastructure of existing research trials in New Zealand, Australia, globally, that we can access. It's like a living laboratory. And importantly, Pinus radiata is also important from a conservation perspective. It is listed on the IUCN Red List in its native range in California and Mexico. And it is one of these plants which is a treasure to the indigenous people in those ranges. So our understanding and advancing our understanding of Pinus radiata for our purposes hopefully can help protect it back in its home range. So we have two very complementary research programs underway. One of these, the tree root microbiome program, is exactly that. It is focusing on the root, rhizoplane, rhizosphere, and soil microbiome of Pinus radiata. Sitting alongside this program is called the Resilient Forest Program, and this program is targeted towards the above ground tissues, including needles, wood, and pollen. For each of the tissue types that we are exploring amongst both of these programs, we are undertaking quite deeply structured sampling strategies. So we can cover where we would collect the tissues from the host plant, so what position in the canopy, height in the canopy, how many samples that we need to take to cover, get coverage of the microbiome in that tissue compartment. We're looking at things such as cardinal direction to see if they make an influence, the age of the tissue, and even how we handle these and bulk them before the lab, in the lab, and at the PCR steps. And all of this is really trying to make sure that it enables us to determine where the key aspects of biological and technical variation are. And it also guides us to ongoing sampling and analysis such that we have confidence that we have reproducible depth of coverage and a really proper compositional view of the symbiont communities that are present. So we're investing a lot of time making sure that the samples that we collect really do represent the microbiome of that tree and not just a a random point in time of a certain needle or piece of wood. So what are we doing with this information? An example of the concepts that we are testing extend from some of those that were originally put forward by Darwin himself a few centuries back, and that is that the root is akin to the brain of the plant, a sensory organ that can enable response and learning from the environment. So we're aiming to try to program the soil microbiome via environmental conditioning, or by transplanting microbiomes from drought-exposed environments into non-exposed environments. We're then going to determine if this knowledge that is held within the microbiome can be transmitted to the plant root, learnt from the plant, and the plant can then change its phenotype and adapt to those conditions. To achieve this, we have assembled a microbiome and metabolomics team here in New Zealand, and we're working very closely with the New Zealand Biotron facility, where we can sample using laparoscopes that go into soil and collect pieces of root tissue in real time, the communication and signaling events that happen at the soil root interface. Systems of interest will be repeated by members of the team at the Phenomics Centre in Adelaide, Australia, and over there they can enable high resolution measurement of phenotype outcomes from the host by environment interactions. Some of the other work that we're doing is testing and developing the rules of assembly of the microbiome on Pinus radiata. And here we're hoping to use the efforts and laboratories of the good folk globally to help us determine the factors that drive assembly of all the microbes in Pinus radiata. So pine is truly an international tree. It is grown in many soil types under a broad range of environmental conditions and across many different management and silvicultural systems. And we hope that the global scientific community can help collect, contribute samples of roots, needles, and soil, and that we can use this to build a natural range of pine as our living laboratory and establish a really robust tree microbiome model. We can use this data set to then start to petition the drivers of environmental influence on microbiome assembly, and perhaps resolve the microbiome of conditionally important taxa, i.e. those that are essential to the survival of pine under quite stressful environmental conditions. Another area that we're very keen on pursuing is looking at the genetic influence in the host plant on microbiome recruitment. So again, we aim to use extensive genetic resources and breeding trials that are already underway in both New Zealand and Australia as opportunities to explore host genetic drivers of microbiome assembly. And of course, this is incredibly complex, with the total phenotype P there being a combination of the genetic, environmental, microbiome, and all the interactions therein. And all of this, of course, sits against an unknown background of random effects and associations. However, and it is a big however, we think that we have the right trials in place and tools to make some really good progress on understanding how much of the microbiome is under some level of host genetic control. And, unraveling this would be wonderful. It would be a brilliant start towards determining if there are breeding values and opportunities for plants, to breed in microbiome associations or select them for the future. And just finally, what have we got there? Range shift and climate migration. So from its home range in California and Mexico, Pinus radiata has now spread via assisted migration nearly globally. Its phenotypic plasticity is absolutely amazing. And there are questions regarding the nature of this plasticity and what its limits actually are. How much role does the microbiome play in this? Has Pinus radiata done so well because it's escaped a heavy burden of adaptive pathogens present in its home range? Or perhaps pine is microbially promiscuous, able to find and form the right microbial associations it needs from nearly all environmental situations. Regardless, this information is really going to help us understand why pine has done so well globally, opportunities for extending its plasticity and phenotype into the future, and also may shed some light on why it is on the IUCN Red List as an endangered species back in California and Mexico. And our program is quite large, it's quite new, it's just getting off the ground. And I'm very grateful for the support that we do have of an international team of experts from University of Western Sydney through to Victoria University in Wellington, Lincoln University down here in Christchurch, the Australian Plant Phenomics Facility, Wright State University, Woodwell Climate Research Centre, and of course Scion. And we're very lucky to have funding from the New Zealand's Ministry of Business, Innovation, Employment, and the New Zealand Forest Growers Research Levy Trust. And with that, I will thank you all for paying attention and for offering me the chance to talk about our new tree root microbiome program to you all. Thank you.
Thank you very much, Steve, for a very fascinating presentation. I'm happy to, like, it's the starting it, and it's something that will continue on. So that's it's going to give us time to see more and more of it. I'm very curious about it. Now we have 1 minute left for a question, and we have a question from Enrico Bonello. He says, my hunch is that a lot of this process are stochastic. What's your prediction?
Yeah, our hypothesis is that a lot of it will be stochastic as well, but there will be some deterministic drivers, perhaps in the conditionally rare microbiome. Certainly, there'll be a lot of opportunistic microbes that are in the environment moving around that can colonize on a plant tissue and exist there. Whether they are altering the plant, altering the phenotype or not is, a matter of question. Really, it's trying to pass all that information out so we can start to understand what the core microbiome is, what are the ones that may have vertical transmission, even horizontal transmission through cuttings, and the microbiomes that are important, conditionally rare, but important under certain environmental situations. So to do this, we need a lot of data, and we need the right type of information from all around the world. And what we'd like to do, New Zealand has a climate at the moment, but with climate change, where we're going is going to be more like Australia and more like Portugal and more like South Africa. So those places already exist, and Pinus radiata is already there in those places. So by heading out globally, we can start to understand what those microbial associations are needed that we can bring back to New Zealand or adapt in New Zealand to protect the pine that we have here. So it's a great question. We don't know the answer. And that's why it's kind of exciting.
Well, thank you very much. Richard, I have a question, but I think it's about time to switch to the full panel discussion. So I'll pass it to Richard, who's gonna moderate the discussion part. And Richard, perhaps you can go ahead and ask that question directly to the whole panel. So I will ask George and Kathrin to unmute themselves so that we can all participate as a panel to the discussion.
First of all, great talks, everybody, and also thanks for everybody that joined in and those that are asking questions. Please continue to ask questions either in the chat or maybe we'll do some verbal too. But I have a bunch of questions, but one I just thought of it after hearing these three talks is, do we need to better characterize what is out there in terms of endophytic communities and natural populations of these tree species we're talking about at different stages in a different environment? Rather than focus down to working with little subsets and stuff like that. What already exists out there? What combinations? How does it vary over the landscape? How does it vary over that? How much of that do we know? And should that be our starting point? That's for all three of you. And anybody else?
I'll start.
Sorry, go ahead, Steve. Sorry.
That's all right. It was going to happen. We've deliberately started working in very simplified forest systems. So in New Zealand, most of our planted forests are Pinus radiata. 90% is a single species. It's under immense potential disruption if a new pest or disease comes into our forests. But also it's being a very simplified system offers to us quite an easy way to get in to start to do the microbiome work, because it's, you know, single species, normally a single age class, and quite basic silvicultural regimes that are happening. So learning baby steps, if you like, on Pinus radiata, and then moving out to other pine species would be the next level of complexity that would be interested in doing. But at the end of the day, you know, we're interested in saving complex forest systems that are mixed species with mixed traits and groups and all the rest of it. And that's a big step for us. So you've hit the nail on the head.
Have you characterized even within New Zealand without their different age within these Pinus radiata plantings, and how that might contrast with what's in the natural range of Pinus radiata as well too? So do you have more diversity of endophytic? Do you have different communities than they do in California?
Yep, at this stage we don't know. One of the first milestones of our project was to start comparing the microbiome of Pinus radiata in New Zealand and Australia back with those populations in Monterey and Mexico, and find out if there has been erosion or loss of the microbiomes as the plants have been migrated around the world. We haven't been able to do that with COVID restrictions. But certainly that's as soon as the people are vaccinated and we're able to travel again, that's our first contracted goal for the program is to do that work. So yeah, it's a great question.
George or Kathrin, any comments on characterizing what's already out there?
Yes, please, George.
I was wondering, of course, about seed, and in particular seed of Pinus radiata in California. And also a comment, I've always thought that the Southern Hemisphere performance of radiata pine was in part about enemy release, because in California, there are a couple of rest fungi and there are a couple of dwarf mistletoes. And so there are fairly strong pathogen pressures on radiata pine in California.
And we can see in our system, the Pinus sylvestris, it's there are more invasion species coming in, and so I think it's really crucial to understand more about the microbiome, also in regard to climate change scenarios. So which species are working together? Is it really just pathogen we are dealing with, or other effects working all together? I think it's being aware what may come in, how are they invading, in which area. So I think all this will be really important to know in future. So we have focused on more of those microbiome systems also in our research, not only to pine, also other species. I think it can tell us a lot what other communities, and what are we dealing with in detail.
What lessons, there's a lot of probably mega work going on with human biota. What lessons are learned from there that can help really jumpstart the work with plants, and what lessons maybe aren't applicable and stuff like that too? As I understand it, we almost wouldn't be alive if we didn't have our microbiota. And so with all the work going on there, how can we capitalize on that and really jumpstart the work with forest trees and other plants?
I think one of the easy ways, one of the first cabs off the block, I suppose, would be the understanding of the gut-brain connections that are occurring. So the microbiome in the gut being in direct communication and signaling with the brain. So there's kind of that reciprocal communication system happening up there, and that is very, very analogous to the root plant microbiome interaction with those signaling processes. The structuring of the gut with the villi, the chemical and redox gradients, the knowledge now that the gut microbiome is a whole lot more than simply a place where nutrients are absorbed and recycled and moved around, but it's a place of signaling and signal modification and dampening and amplitude and all the rest of it that connects with the rest of the plant. And those are wonderful analogies that we can take into the plant root microbiome system to start looking at the microbiome involved in the root. So what's happening in the root is really in communication with the rest of the plant through similar signaling pathways. And some of those are quite simple based on sugars, and others are a lot more complex. But yeah, there's a lot we can take from the human model.
I can think of one particular study, and this was a group of Parkinson's researchers, and they were using C. elegans as their model organism. It had been genetically engineered to produce the toxic protein in Parkinson's. So this is alpha-synuclein. And then they were using various gut microbes to see whether any of the gut microbes could shut down the production of alpha-synuclein, which is produced in the gut, but then travels up the vagus nerve to the brain. And Bacillus subtilis was the most efficacious gut microbe. And again, it highlights something that's exciting to me, and that's the very strong antagonistic ability of Bacillus species.
Yeah, but talking about the differences now to plants, I mean, in such systems, we have an idea where it's taking place. But transferring this to the plant microbiome interactions, I think that is still one of the big obstacles for us to understand where are those interactions happening. So maybe we can engineer certain microbiomes, but how can we control that the actual fungi of interest meet the pathogens or where they are supposed to be? And yeah, so this is something I find really also interesting to take into consideration. What's your opinion here? What are our chances of getting into that directions?
I'm not sure who you're asking, but
so when we find out, when we do the studies about the microbiomes and understanding better what are we dealing with, so what does this give us in the end for information? How we can we use this? Maybe we know, okay, that's in a certain organ of a plant, and our pathogen is attacking from a certain type. So how can we make this information of use? Bring into transaction.
And maybe that is reflecting back on the human microbiome model. So that started off as a curiosity project to catalogue the bacteria and fungi and other organisms that are existing in our bodies and different parts of our bodies, and has now reached the stage where, you know, we're completely rethinking what it is to be a human. We're not just a collection of human cells, we're a collection of human and microbial cells that are acting as a meta-organism. So it's changing some of our ecological theory and actual way that we are holding a mirror up and looking at ourselves and how we function. We understand that the microbes that are on and in us are in that kind of balance where, you know, if our diet is wrong, then they're upsetting our brain and brain signaling. And if our stress is wrong, where it's upsetting our diet and all the rest of it, there's all those sorts of balance, which is affecting our fitness and our health and our longevity. 90% of human diseases are thought to have some sort of microbiome association somehow linked to them, whether that's right or wrong. It's going to be a very high number. And I think perhaps some of the really interesting stuff is by doing this work, and you don't know what's going to come out, all these unexpected and exciting new areas open up, such as the role of the microbiome in Parkinson's and Alzheimer's and chronic fatigue syndrome and all these other psychophysiological conditions, because of that connection between the gut and the brain. And if 10% of those sorts of wonderful new things occur in plants as they do in people, then it just opens up whole new avenues of opportunities for us to start to understand plants better, reshape our thinking around what a plant is in the environment. It's not just a collection of plant cells, but it's a whole assemblage of a plant and the microbes, which may change over time as well. So yeah, just changing the way that we think about plants and forests into that whole meta-organism holobiome theory, I think is a really, really exciting place.
I think too with the human microbiome that there still aren't really good applications, and probiotics are not really state of the art.
Just shows how much we have to learn, and it'll be great to have a group of people get together and put together a roadmap for, there's already a good start there, but it's a little bit piecemeal and whatever too. But if you had the resources and you had, what would you do over the next 10 years? And what needs to be done over the next 10 years and whatever like that, almost on a parallel track with the human. I think we're going to switch, and Caterina is going to ask a couple of questions that are in the chat box, and I think we have about 5 minutes left. But the roadmap would be a really good thing.
We had some about 4 or 5 questions left from Catherine's talk, and one more for George. But I think we could send those out by email because they're specific for your talks, and I will probably go ahead and ask. There are some questions for the whole group, and I know that Enrico has his hands up as well for a question. I'll start with Todd Rensfield question, which is for the whole group. How would the advent of next generation sequencing method change the ability to characterize the microbiome versus traditional culture-based methods that may miss known cultivar species? Next generation methods often result in detection of a very diverse community compared with culture-based methods, but I wonder about the reliability of those next gen data sets as they seem almost overwhelming. What are your thoughts?
I agree. I agree with the sentiments expressed by Todd.
Yeah, me too. So we're increasingly taking complementary approaches where we're spending a lot of time and money on sequencing and discovering potentially unknown or spurious maybe taxa in our samples with no idea around their ecological role or significance outside that they exist in a certain space and time. So spending quite a bit of time now trying to actually get those organisms back into culture and finding out what their genomes are, and then being able to start to shed some light on the ecological dark matter around what it might be doing in terms of its association with the plant. So the more sequencing that we're doing, the more next generation sequencing we're doing, is actually showing that we need a lot more fundamental basic knowledge on the actual organisms that are in these systems.
Thanks. Enrico, do you want to ask your question?
Yeah, thank you all, guys. I have one comment and then one consideration. The comment is that we need to be careful using human microbiome as a potential roadmap or a reference for what you try to do in ecological systems. Humans are just one species across the globe, which is why we have pandemics, but we don't have pandemics of trees because trees are discretely distributed around the globe, usually on subcontinental scales, for example. So there's a lot much more variability. And so that's just one consideration. The other one is, maybe this is a stupid concept, but I'm thinking if we can take more of an ecological / evolutionary look at what goes on between endophytic communities and pathogens. If you take any one tree population, let's take one population of Pinus radiata on the California coast, and you ask, are there endophytes there that could be helpful in protecting the trees against certain pathogens, which are already resident in the area? Wouldn't you expect, to some extent, that natural selection would have already selected for the best endophytic communities to cope with those specific pathogens? In other words, we can analyze the whole endophytic community and then do some experiments and figure out which ones of those microbes seem to be the best in controlling a specific pathogen. But the overall community is affected by the push and pull of natural selection. So in a way, my question, I suppose, is what's the point if everything gets washed out in the not serendipity, but I don't know what the word is, in the ecology of the system, I suppose.
In my view, I think natural selection in a system or in a scenario like that is complicated by the bottleneck that the seed represents, because the endophytes that might be interacting with a rust or a dwarf mistletoe in California would have to actually get into the seed. And that means being able to exclude other seed infecting endophytes. And that's a totally different set of abilities from what may be required to effectively interact with the mistletoe or the rust.
Yeah, but if you look at a tree as a composite organism and individual branches, for example, on a tree as separate communities, rather than having to go through the bottleneck of the seed, which I understand, I understand your point very well. This is an open question. I mean, I don't have an answer. I mean, I'm just curious to see how you guys think about this kind of stuff. If a tree is a sort of a modular organism with different parts that may be infected with different endophytes, would you expect perhaps different selection pressures on different parts of the tree? And I mean, I'm thinking out loud here, okay? I'm out on a limb, no pun intended. But I think these are maybe interesting questions to ponder.
I think Caterina can let us know how much time we have left, and we're probably going over, but just one thought. It seems like, a lot of things are being done on a relatively small scale, that more, whether an IUFRO working group, a task force, or whatever, to really get at, what role does the microbiome in trees play? How should it be harnessed? Can it be harnessed? Et cetera. Some very basic questions.
Yeah, so we'll have to start wrapping up. We are just about at the end of the discussion, but there were many unanswered questions in the chat that we will make sure to send out via email so that we can keep the discussion going on. But I would like to thank you all for participating. A special thank to our 3 speakers today. Great talks, very, very interesting and thought-provoking, which is the nice part of it, that gave rise to a very nice discussion. And please keep following all the series of this new webinar series every other Wednesday. We will send out reminders on a regular basis so that you have the link and you don't forget to log in. And again, thank you all for participating.
Thank you.
Yeah, I'm seeing a lot of questions and comments. We will make sure to put them like in written and send them out to speakers for sure, and probably to the list of attendants so that they have it. And thank you all.
Thanks, Caterina. Thanks, colleagues.
Thank you.
Take care.
Thank you very much.
Thank you.
Bye.
Thank you, organisers.
Bye.
Thank you, everyone.
Bye.